right? So it's going to be over here, unwinding that DNA strand as the fork breaks open. Now there's another one. What's the zipper one called?
Helicase.
Right, and what's the helicase? What's she do?
It uses ATP to break hydrogen bonds between the strands.
Whoa, look at you, scientist.
Someone's making it after me.
All right, you didn't have to ruin it for me.
I said almost.
So the helicase uses the ATP to break, and this is important, hydrogen bonds. Oh my God, I cannot write today. Just ignore that. That was disgusting. Hydrogen bonds. Cool. So that's an important thing that I want to remember about a DNA molecule. The DNA molecule is made of two DNA strands, and they're held together by hydrogen bonds. So if I were to break all the hydrogen bonds in a DNA molecule, I would have resulting 2 DNA strands that are separated from one another. That's what the helicase essentially does, though, a little bit at a time. It breaks those hydrogen bonds. Along with the helicase, there's these little doodads over here at the top and the bottom. What are those little doodads, and what do they do? Somebody raise their hand and tell me this one. It's getting crazy with all this shouting out. Just because, not that it's a problem, it's just like, it's hard to hear my brain. Brianna.
They're binding proteins.
They are. Yeah, they hold it open, that's right, so that it doesn't accidentally close back in on itself and reform those hydrogen bonds. Cool. All right, so let's talk about a really important molecule here. Right there. Here's one. There you go. So this really important molecule, it comes in a couple varieties. Now, I don't want to know what the name of this one is from your paper. What I want to know is something else. It has a general name. Well, kind of a general name. Let me rephrase the question. What do we call an enzyme that is designed to read a nucleic acid and write nucleotide bases to that nucleic acid? It has a specific name. You can change the first part of the name then or add like a prefix to it, but what do we call that particular type of enzyme? Again, it's an enzyme that reads a nucleic acid like DNA or RNA, and it will write nucleic acids to that particular nucleic acid. Yes?
Polymerase?
You got it. It's a polymerase. That's it. It's a polymerase. Polymerase. Any molecule or any enzyme that reads the DNA or RNA strand and it writes subsequent nucleotide bases to it is going to be called a polymerase. In this case, because it's DNA replication, this one is going to be called DNA polymerase because it's writing DNA nucleotide bases to the DNA strand. Okay? Now remember, the DNA polymerase, it is very specific in how it reads and writes. So when it's reading a DNA strand, which direction does it go? 3 to 5. It's going to read from 3 to 5. Okay, so that's reading. And which direction then is it going to be writing the DNA strand? It's the opposite. You got it. It's going to be writing from 5 to 3. And so essentially what that means is that as it's starting at one end, it's going to be reading the DNA strand. The DNA strand that it's reading is going to be from 3 to 5. And as it's writing those nucleotide bases onto it, those nucleotide bases are going to start at the 5, and they're going to be working towards the 3. So it's not like it does 2 different jobs. As it's doing one job, it's doing the other one at the same time. So it's reading from 3 to 5, but it's writing the new DNA strand from 5 to 3. So, and then at the very end, I end up with another full DNA molecule. So let me shrink this down just a little bit. Hold on. Let me get him out of the way. He's in my way. So we can get all those. There we go. Sweet. Cool. Let's take a look at this a little bit more closely now. So as I was just describing with the polymerase, it reads in a 3 to 5 and writes in a 5 to 3. So because of that, I end up having 2 different strands being written at the same time at the bubble of the DNA or at the replication fork. So here's my polymerases here, the little pink guys, the little pink doodads. As they're reading and writing, what happens is because of the different directions that they're going, it occurs bidirectionally. We have what's called a leading strand and a lagging strand. So here's my top. Here's my bottom. Take a look at this diagram very closely and try to determine which is the leading strand and which is the lagging strand, top or bottom. Think about it. Remember, the polymerase reads from 3 to 5 and writes from 5 to 3. The leading strand is going to be the one that follows the replication fork all the way. The lagging strand is going to be the one that has to go in the opposite direction. So take a look at this diagram, see if you can figure out which is which. Any guesses? It's kind of a tricky one. Brianna?
The top is the leading strand because it starts with a 3.
Perfect. Top is my leading strand because it starts with 3. And as you can see, I've got my 3 open here. So this is where the polymerase is going to bind onto this old DNA strand and read in that direction. And then it's going to be writing, starting with the 5 to the 3, going in that direction. As the replication fork opens up further upstream, this will be able to follow it all the way down without having to break off. Down here, though, the bottom, that's going to be my lagging strand. And it's lagging because the 3' end that it needs to start at is actually still caught up here in this tangle. It can't get to it. So the closest it can get to is like here. So it's going to start here and it's going to read from 3 to 5, writing from 5 to 3. Then when it gets to the end, it'll jump off. It'll go further upstream where this will hopefully be untangled and it'll be able to continue on until the spot where it started beforehand. So this is my lagging strand at the bottom, leading strand at the top. And remember, it occurs bidirectionally. So this one is going in that direction, this one's going in that direction. And ultimately, I've got lots of these little replication bubbles, just like I've got shown right there in that diagram, that occur all over the DNA strand to make it happen really, really quick. Okay? Question, what phase of mitosis, what phase of the cell cycle does this process happen in?
S2.
S phase. S phase, because remember, S stands for?
Synthesis.
That's it. Synthesis. Synthesis of the DNA. So I'm going to be seeing replication happen during the S phase of mitosis so that the DNA can be totally synthesized and replicated, and boom, ready to go for mitosis. Cool. All right, got that down pretty well. All right, let's model transcription real quick. So here is my RNA polymerase. There we go. Here's my DNA strand. Remember, as I mentioned before, polymerase is any molecule that's going to read a nucleic acid and write subsequent nucleotide bases to the corresponding nucleic acid bases. In this case, with transcription, since that was replication, transcription is basically taking the DNA strand and turning it into an RNA strand. Question, which type of RNA is it going to be turning it into?
mRNA.
mRNA, good job. That's sign language mRNA. It's the only one I know. Actually, I know N too. N. Got it. Nailed it. All right. So when I'm taking this polymerase and I'm cruising on up the DNA strand, am I copying the entire DNA strand? No. What am I copying? The what?
The part you need, I guess.
The part you need, sure. The part you need. In this case, it's going to be a specific gene, right? So in this case, I've got this gene ATCG right there. So I'm going to cruise my polymerase along this line right there, just in this section, and I'm going to be matching them up with the corresponding base pairs. So A with T, and U with A, and C with G. Whoops, I don't know why I did that in that order. It doesn't matter. And then there we go. Cool, cool. And so there's my mRNA strand. Awesome. Bring that up there. Awesome. So I've got my mRNA strand ready to go. Now, question. In a eukaryotic organism, like me and you, does it go straight to, what's that organelle called again where translation happens? Where I make the proteins? What's that organelle called? Begins with an R and rhymes with ribosome. That's it. I didn't hear it. It's okay. No, get mad at me. That's fair. So anyway, so I've got the mRNA strand. Oh, Amber. I got the mRNA strand, and it's going to go to the ribosome. But does it go directly to the ribosome in a eukaryotic organism?
No.
Something happens in between, right? What happens in between? Something happens before it gets to the ribosome. And I don't remember what it is, but I know it's written in my notes someplace. So I'm going to take a look at them real quick.
Your notes.
Yeah, I think it's on the back side. I think it's letter C. I think it's number 2, but I'm not 100%. So double check there for me. See if it says anything there. What are some important things that Wait, what?
Enzyme regulated modifications.
Yes, thank you. Enzyme regulated modifications for sure. I get some enzyme regulated modifications. So what's going to happen here is this mRNA, it has some parts in it that aren't necessarily needed. And so if they're not needed, we're just going to go ahead and splice those out. So like for instance, I don't need this portion. So I'm going to cut that out and just connect those together. Boom. There's my new mRNA strand. What do I call that process, by the way, if I'm taking out parts that aren't needed? I think that's also written in there too. It's one of the bullet points. That's it. Excision, I know I spelled it wrong, of introns. Translation. removal of non-coding regions. That's what that means. Excision of introns means removal of non-coding regions. So I've got an mRNA strand, and it's got a lot of different things in there, some of which are important and others are not so important. So the, what did you call it again, Amber? Enzyme something?
Enzyme-regulated modifications.
That's it, enzyme-regulated modifications. In those enzyme-regulated modifications, one thing that happens is the excision of introns, or the removal of non-coding regions. And then it can go to the ribosome, and then it can do its thing. See, I think I used the analogy before where you're making the copy of the encyclopedia at the library, and you're just copying a small portion first, and then there's other things in that portion that you don't actually need, like diagrams, so you cut those out before you finish it. Yes?
So how is this part different from RNAi?
It's related to the RNAi, because the RNAi is also regulation, but another thing about RNAi is also another grouping of things that happen after the mRNA is done being translated into proteins, where the RNAi also comes in and not just, like, it doesn't just take out introns, it actually cuts the whole thing up into pieces. Okay. So the RNAi is actually this whole process of splicing the mRNA and chopping it up. And a lot of times the RNAi can actually be used to control gene flow, where an mRNA might be transcribed, but it never makes it to the ribosome because the RNAi comes in and just chops the whole thing up before it actually turns into something. And we'll learn more about that when we get to 3b. And gene regulation. Okay, cool with that, yeah? Transcription, awesome. All right, let's talk about translation. What was the name of that molecule, that organelle again, where the translation takes place?
Ribosome.
The ribosome, good job. Good job knowing that. Definitely happens at the ribosome, but there's a lot of other things that happen that's not shown in this diagram here, so I'm just going to draw it out real quick, and you can just watch me do this art. Well, actually, it's not art, because I'm not going to label it, so it doesn't actually exist as art. Here we go with my ribosome right there. Okay, so let's talk about a couple things. So here's my ribosome. Here's my mRNA strand. Well, let's just start with something simple. What are these, what is this called when I'm like, what is the...
Start codon.
I heard the C, the codon, yes, that's what I'm looking for, codon for sure. I was trying to think of a way to like cleverly say like, what would you call the word in the, I can't even say it in the fake voice. So that's a codon, 3-letter sequence, that's going to code for a particular, what is this code for again? No, not this in particular, I mean in general, like here, this one, same thing, you know, this, every 3 letters is a codon, and it codes for a what?
Amino acid.
An amino acid, yes, I heard. An amino acid. It's going to code for a specific amino acid. Let's talk about this first one again. I heard somebody say start codon. Is there a particular codon that's, what is it? AUG. That's right. So this is like the capital letter of the sentence, AUG. That's going to tell my ribosome this is where I need to start reading this mRNA strand. And it's also going to code for its own particular amino acids. 3, there we go. All the way down. So I've got those 3, that codon there. The codon codes for a specific amino acid, but how do I get what transports the amino acid to the ribosome? I heard it whispered on the wind. It's the tRNA. There it is, the tRNA. It's got that little amino acid attached to it right there. Amino acid, perfect. Now, if the codon is on the mRNA strand, what is on the tRNA strand? What are those 3? That's it, the anticodon. So, on the mRNA strand, I have the 3-letter codes. They're called codons, and they code for a specific amino acid. On the tRNA strand, it's the exact opposite of those 3, and that's called the anticodon. So, if I was to draw it out, say, like, here's my mRNA strand, and it has A, C, and then G right there. Here's my tRNA molecule, and here's the anticodon. What would the anticodon be for these 3 letters here? What would that one be? Well, that first one. Let me repeat the question again. This is my mRNA strand. Here's my tRNA. Here's my codon on the mRNA strand. So what would my anticodon be? What's this first one going to be?
U.
What? It's RNA. So
U.
That's it. We got a U. How about this?
G.
W, G, and C. Perfect. There we go. So this would be my anticodon. Anticodon. This would be my codon, and this, of course, is the tRNA. Which, by the way, what does the T stand for in tRNA? Transfer, transferring the amino acid, and then of course this is M. RNA. Okay, so there's my codon, anticodon thing going on here. And then of course the amino acid gets dropped off and it attaches to the previous one, and that attaches to the previous one, and that's attached to the previous one. And eventually I get this long chain of amino acids. What do I call a long chain of amino acids?
Polypeptide.
Right, a polypeptide, or what's the layman's term for it? Protein. Polypeptide is a protein. Polypeptide and protein are one in the same. They are synonyms. Okay. See if there's anything else. Oh, almost forgot. How does the ribosome know when to stop reading it? Yeah. There's a stop codon. We didn't learn which one in particular, but there's a couple different ones, or I should say there's a few. There's 3 different ones. There's 3 different stop codons. And once that stop codon is reached in the ribosome, that terminal stop codon basically shuts the whole process down and it's done. The polypeptide detaches from the ribosome, and then of course that polypeptide goes and does whatever it is it's supposed to be doing. Since it's a protein, it has some sort of a job, whether it be an enzyme or a building block or whatever, but that is essentially how genes get turned into actual physical whatevers. So like, you've got your DNA, that's got your genes on it. The genes get read by the RNA polymerase, which turns into mRNA, and then the mRNA goes to the ribosome to get turned into protein. And then that protein does something. Because your genes are slightly different than my genes, your proteins are going to be slightly different than my proteins, and therefore you are going to look and behave differently from me. That's how that works. That's how genetics comes into play. Additionally, as we'll talk about a little bit later on, remember inside your cell you have basically 2 chromosomes, one from mom, one from dad. And that whole allele thing, dominant, recessive, that's going to determine which one of these chromosomes ends up getting read. So like for instance, if your mom has a dominant gene for brown eyes and your dad gave you the recessive gene for blue eyes, this is the gene that's going to be read, not this one. And so this is where your brown eyes are going to come from. Even though you have this on your chromosomes, your body never actually turns it into protein via this process that we just described. It's just brown eyes all the way. Cool?
Yes.
Awesome. All right. Do you have your active expressions? No. You don't, but come on up and get them because we're going to be using them now for some questions.
Oh, that's why you're more like. Yeah, you can send it if you can. Yeah. When I hear what I do. I'm not doing no. I'm just like even if I have it, I should have stopped.
Wait, where'd you go real second? Real This is what? Some people laugh. Annie laughs.
I actually thought I would not go but I still have not gotten it.
It works.
They're small.
Okay. So here are going to be some questions for you on your active expression. I know. Here's going to be some questions for you on your active expression, reviewing some of the things we just talked about, but also reviewing some of the things we're about to talk about, like mitosis, meiosis, etc. As always, work with a partner, work with a buddy, because biology and buddy start out the same way.
Oh, that's the only reason why.
Right? No? You got a smile from Tracy. That's good enough. All right. Work with your pal.
What does I say? Interface. Interesting.
All right. Ready, set, go.
I have biology.
Mine isn't showing anything.
It's showing nothing.
Or like it has that little band.
Try like pressing the circle button a whole bunch, just rotating around the circle button.
What are we doing?
Anything? No? Here, I'll trade you. Yeah, 00. You're just like dying. They get pretty dramatic. Oh yeah, it is. Yours too.
Oh, so if it's cut into 4 at 4, then we have 5.
Made of batteries.
Yes, they are.
Versus
Not magnetic, it's just attached. See?
experiments in the percentage of adenine in the DNA proportion of guanine.
It is experimentally determined.
Some of these questions may or may not find their way onto your test on Monday, just FYI.
In the rest.
You calling me lazy? Saying I wouldn't come up with my own new questions.
What? Oh, so 30.
One time I almost broke it in a
Read your math. When molecules of hydrogen bonds break readily, how will this affect DNA if it is heated?
So that's a headphone bill. Wait, hold on. Wait, so it use the original headphone jack even though? Done. Done with their pass.
Where are the hydrogen bonds? There's no H.
Only good?
Then it will.
Grace good?
Yes. No.
Guys doing okay?
Why am I so bad?
He's good over here?
I feel like I
Here, you guys come back. I think she had surgery. I think so. But she's okay. You guys good? You guys good back here?
Yep.
Is it okay? You just have this chill in here? Because I know you're gonna accidentally walk out with it, and then I'll never see it again.
Okay, hydrogen.
Oh, yeah.
So,
Yeah. Oh, wait.
Are you actually gonna watch it later?
Yeah. Oh, on Sunday. Is that okay?
That's totally fine.
Okay.
It's, I mean, it's late, a little bit late now. You've got like 20 minutes of me or whatever. 40 minutes of me. So
Well, when did you start? You started filming.
I have 23 minutes.
that's fine.
Are we going over these questions?
We can. If you have particular ones that you want to, I'll bring them up.
Okay.
Last class didn't want to go over. So I was like, all right.
Why not?
Because it's last class. They don't want to do anything.
Are you gonna fill out the rest of the?
yeah.
Oh, the acrostic? I can.
Outstanding. Logical.
Let him
Overachieving.
Sounds like you've already got it.
Why?
Yes.
Yay. What is the AP of E? Okay. I should have looked for you. Okay, 6. I think it was 5. Or no, 4.
Good.
What? Oh my god.
It was terrible. It was really funny.
No, it was the first one only.
Oh, I can't read. Wait. Mr. Arnold, I can't read the rest of this answer.
Let me see.
Oh,
What the heck? Ooh, that is annoying.
Maybe it's supposed to be a dot.
Yeah, that's weird. I think it is definitely all three of these.
Yeah.
I just don't know why it won't. I can bring that one up and look at what the answer's supposed to be. Yeah, I'm not sure what's wrong with number 8 and why it's only showing. See what I mean? Last period didn't even tell me that.
Oh.
They didn't even mention that to me. This is the first time I'm hearing
Do
it.
they just not like me?
Huh?
Maybe they're
Did
asleep.
they just
They're
not
just
like
asleep
me?
during the
I think it's a combination of both of those things.
well.
And it's first. It is all three. Also, it's just first period, so they might just like sleep.
It's got a lot of people in there who don't actually want to be an AP Bio.
Why do they take the class?
That's an excellent question.
They just get an open period. It's a first period.
Yep. Now, there's plenty of people in there who do try, who enjoy the class, but there's lots of others who just exist. Barely, too. Like, barely exist.
But every time I come in, I get a little bit more concerned now.
Yeah,
I
and
do
me. All four.
I'm working on it.
Then I go home. Like I read it all and like damn, I'm so good.
That's because, like,
It's the last page.
you can pretty much say whatever you want. Get away with it. About 45 seconds. If there's a question that you want to discuss or talk about, I will bring them up afterwards. We'll talk about those. Every single one. We'll definitely talk about a lot of them. Some of them we will be reviewing the how to get those answers a little bit later on too, though.
Okay, every answer on question number 6 is wrong.
Are you sure?
Because I've tried them all.
What's number 6?
I got 6 wrong too.
Which is not a key step in the process of translation.
It's C.
I put C and it was wrong. Okay, now it worked.
Okay.
I had tried 6.
We're gonna look at 6 for sure. We can look at some of the other ones too.
I didn't let me answer the rest of
Combo breaker. Hey, would it be helpful if I posted those questions on Classroom?
Yes.
I'll do that for sure. Let's take a look at some of them though right now. See if that's okay with you. We're gonna look at a lot of them. I don't know if we need to do all of them because we still have some reviewing to do after this too. We'll see how we go. Okay. Let's look at number 6 first, which is not a key step in the process of translation. A lot of people got this one mixed up, whatever. If you read closely at letter C, and then you can't see, it says the sequence of nucleotides on the tRNA is read in triplets called codons. What is actually read through the ribosome? Which molecule of RNA? Which type? It's the mRNA. This one says tRNA is read in triplets called codons. The tRNA contains the anticodons and it brings the amino acids to the ribosome. So that one is the one that's not correct for this one. Kind of a trick question there. Not really super fair, but that's what you're looking for. Okay, something simple like that. For the other one, Benedict, which one was it? 8. It says prokaryotes produce proteins faster than eukaryotes. What makes this possible? Select any and all that apply. There were 3 answers. All 3 of them were correct. It should have said prokaryotes do not modify the mRNA post-transcription, so that excision of introns. It says prokaryotes do not have introns to splice out as eukaryotes do, so kind of the same thing there. And then finally, transcription and translation are coupled due to a lack of nuclear membrane. So remember, in eukaryotes we have a nucleus that contains the DNA, and then the mRNA has to travel out of the nucleus to get to the ribosomes. Prokaryotes don't have that feature. So as soon as the mRNA is being produced, it binds onto a ribosome and immediately gets translated. So there's never any downtime between the two. That's why prokaryotes produce it a lot quicker. Were there other questions on here that didn't necessarily have to do with interphase, mitosis, or anything like that? So we're going to review those in just a second. Yes.
Number 4.
Number 4. When a molecule is heated, hydrogen bonds break readily. How will this affect DNA if it is heated? So what holds the 2 DNA strands together in a DNA molecule? It's the hydrogen bonds, correct. So if I were to heat a DNA molecule and it breaks all the hydrogen bonds, yep, it should split open. So I end up with, the DNA will separate into 2 single-stranded sections. Cool.
Got it.
There was another one on here I thought was kind of interesting. Anyone get number 2? It says that bacterial DNA was cut into 4 locations with restriction enzymes. How many separate sections of DNA will result? It should be 4. Now remember, the reason for this is because bacterial DNA is circular. So if I take a circle of DNA and I cut it in 1, 2, 3, 4 places, I get 1, 2, 3, 4 sections.
Oh.
Versus human DNA, if I were to cut it in 1, 2, 3, 4 places, how many sections do I have now? I've got 5. So keep that in mind. Remember, our DNA is linear. So cutting it into those sections is going to result in more sections than I would if I had, say, bacterial DNA, which is circular in the prokaryotes. Okay, so there's that. All right, so let's talk about some other things real quick together. Let's review the cell cycle first. So I'm going to start it, and on that piece of paper with your buddy, look at that circle. That was so. Let's try another one. Oh yeah, that's it. What can I say? I'm pretty good. I'm going to start the circle. You go ahead and finish the cell cycle for me now on your piece of paper. Finish it up. Show all the different parts. I'm going to finish mine up in a minute or 2. Make sure you label everything. Remember, it doesn't exist if it's not labeled. It's not real.
Okay, you can test me.
Yeah.
Fine.
If you're not labeled, you actually exist.
Yeah. Okay, so
You actually
I know
exist.
this right. I know this right here.
this is the S phase.
G1.
Yes.
That's why high school's so
I don't
hard.
remember what
You
this
look
is.
labels quick. Labels quick.
I don't remember
That's
what
right.
What's my label? So the first one is yeah, but there's like division of cell, organelles.
Yeah. Oh, yeah.
So
That was fast.
I remember.
yeah. That's what it's called. Keep my so it's like S phase, G phase, G2 phase is where they make. See, she laughed at it.
What?
MPS.
And then S phase.
I would never.
Person who's gonna kill you definitely would say I would never.
Yeah, in that voice.
If I kill you, I don't kill you person again. So I kind of need you.
That's fine.
They don't do anything.
Why don't
It's not because you're my friend or anything. And like, so it either this part of this section is like sorry, what it actually happens. Both of them.
With the splitting, right?
Yeah.
Yeah.
And then afterwards it'll either go back into interface or it'll just G0
Yes.
just
So interface is
So this is all interface.
All right.
Okay, so this is
So I'm gonna start drawing mine up here. You help me out. I'm gonna divide it up like this.
Oh, yeah, I know.
Checkpoint. Yeah, one is right here.
What is this overall phase from arrow to arrow called?
interface.
You got it.
And then checkpoint somewhere.
Yes.
That's interphase.
I don't
What's
remember.
the other? What's this yellow phase called down here?
Mitosis.
That's right, mitosis. Sometimes also called M phase. I can call it that too. Mitosis also branches off at one point, like after mitosis, I should say. I have the option to kind of branch off to something else. What's that extra secret phase called?
G0.
G what phase?
Zero.
G0 phase, you got it. Or sometimes I call it what?
No.
I call it job phase. Job phase. Remember, that's where the cell's going to do its job. Most cells spend a lot of their time in G0. Very few of them will enter back into interphase going into the mitosis phase again. Like, for instance, nervous cells. Once they've divided once, that's it. They're nerve cells forever. And then that's their job. Same thing with a lot of other cells in your body, too. Some cells, they will go back into interphase if necessary. Like, for instance, if you get a cut on your skin, that skin needs to rebuild itself. So there's going to be hormones released and different enzymes released at that particular area only to let those cells know, hey, you need to start growing so that you can repair this this cut that's here. But remember, that's not a hormone that's going to be released all over the body because that would result in some major issues if every single one of your cells entered into interphase and started dividing rapidly right there. Instead, it's just going to be a localized thing. Most cells are like that. So anyways, going back to the actual interphase now, What's this first section called?
G1.
G1. And what happens in G1 phase? Right, I'm going to see some division of organelles. Division of organelles. And then I'm also going to see some growth happening here too. This phase up here just underneath where I wrote interphase, what's that going to be?
S phase.
You got it, S phase. And what does S stand for?
Synthesis.
You got it, synthesis. And what am I synthesizing during this?
DNA.
DNA synthesis. So I'm going to be replicating the DNA, all that stuff with the topoisomerase and the helicase and everything, that whole synthesis, that whole replication process that we did with the replication fork, that's going to be happening right inside here during S phase. Okay, so by the time S phase is done, I will have double the normal amount of DNA in my cell. And then I spend the next entire phase, what's that one called again?
G2.
G2. What am I doing during G2?
MPF.
Yes, both of those things. I'm going to be prepping for mitosis. Prep for mitosis. Mitosis. And I'm also going to be making MPF. Let's talk about MPF real quick. What does MPF stand for? Well, the M is the same as this. Yep.
Promoting factor.
That's it. Mitosis promoting factor. Who was that?
Me.
Nice job. Mitosis promoting factor. Now remember, mitosis promoting factor has two parts. And those two parts kind of go together like a puzzle piece. One of those parts is always in the cell at all times. From during mitosis all the way through interphase, it's just kind of floating around doing nothing. That part is this bottom portion here. What is it called?
CDK.
Right? It's called CDK. Does anyone remember what CDK stands for?
Cyclin dependent.
That's close. Kinase. Cyclin-dependent kinase. Kinase is another fancy term for an enzyme that does some sort of a job. In this case, a kinase is an enzyme, bless you, that its job is to phosphorylate other enzymes. So what it does is the CDK is there to phosphorylate enzymes or phosphorylate motor proteins to make them do their job during mitosis. That part isn't super important, but the CDK stands for cyclin-dependent kinase. So the kinase is the thing that phosphorylates. Dependent means it needs something else, and cyclin is that thing that it needs. What's this top one called then? That is cyclin, that's right. And cyclin, of course, is named as such because it is produced at only specific times during the cell cycle, namely here during G2, and that's where the MPF is going to be made. So the cyclin makes the CDK be able to work, and then the CDK and the cyclin together makes MPF, which basically powers mitosis. That's going to be what it does. Power mitosis. That's its job. So MPF is going to power the mitosis. If there isn't enough MPF for mitosis to take place, then mitosis will not happen. We're not going to have any division of the cell. And that's, again, one of those things that the checkpoints is at. If you label the checkpoints in there, there should be one at the end of G1. There should be another one at the end of G2. And then there is another one halfway through mitosis. Those are my cell checkpoints. And what their job is to do is make sure that all the things that are necessary for the next step are in place. So after G1, after normal growth and division has happened, it's going to double check everything to make sure that everything is ready to go, because replicating the DNA is kind of a big step. Like, it's no big deal to grow and make more organelles. If you stop at this point and don't do anything else, the cell has still got it's got more organelles, and that's no big deal. And it's a little bit bigger, that's no big deal. But once it replicates the DNA, now you've got 2 copies of the instructions for life. That's going to be an issue if you stop at that point and don't do anything else. So it wants to make sure that everything is in place. Replicate the DNA. Now I have double the amount. At this point, I want to make sure that I've got enough MPF. If there isn't, then I have to stop and produce more cyclin. Then, through mitosis, I'm going to check for MPF one more time, and then I'm going to finish the division. And that's it. And the cell will continue on about its business, either go back into interphase and dividing again, or it will go down into job phase where it will do its job. And then ultimately, at the end of this whole cycle, I end up with 2 identical daughter cells, genetically identical. So they have the exact same DNA and everything else. All right. So let's now talk about the differences between mitosis and meiosis. So here's another drawing for you. Here I've got a normal body cell, which, by the way, what's a normal body cell called?
Somatic.
Somatic, that's right. Somatic cell, that's body cell. This one is going to be undergoing mitosis. And this somatic body cell is going to have 28 chromosomes. And then down here, I've got another cell, but this time it's not somatic, it's not a body cell, this is a gonad cell. Remember, these are the cells that will obviously only turn into sex cells. The somatic cells won't be doing this. This one also starts out with 28 chromosomes, but this one's going to be undergoing meiosis. Right? So from here, you and your buddy take a minute, show me what's going to be happening along these lines. Divide those cells up, get to the end point. If anything important happens during these divisions, label it in there. Okay? Make it like point it out. There are some important things that happen in some of these that we need to talk about, we need to know about. So go ahead and take a minute with your buddy. Label these, finish them up, finish the drawing. I've got one cell undergoing mitosis, one undergoing meiosis. Finish the whole thing, finish the process. I don't need to know the prophase, interphase, and all that other stuff. What I'm talking about is they're going to be divided. I want to know like how they divide. And the number of chromosomes in each one, that's what I want to know.
Aren't you the one that's what?
I'm accepting that.
So it doesn't matter that it's not 46 like that it starts out.
Because this is not one of our body cells. This is a different organism. Yep. Now I'm using a different number so that I can
Don't
see
memorize
that
it.
Yes, you got it. Yeah, so this is a different number because I want you to show me what this particular organism would undergo as far as its chromosomal changes.
Oh, I have this. Oh, I think. I kind of memorized that number. So first, it's gonna replicate all the DNA, right? So we're gonna take it as 56, right? So we're at 56. We're at the bottom.
Yep, that's good. Well, it's kind of like meiosis, except it's fast.
So does that mean like
Yeah, those are all part of it, but I want to make it
Blood pressure.
Yeah, that's what I remember. That's like, wait, oh, okay.
So in both of these, they might start, they double right? Did you know you have high blood pressure too?
Did you know that you were walking? It's basically also.
Okay. So let's take a look at your drawing versus my drawing. Okay, we're going to start with the mitosis one. First thing that's going to happen, I'm going to see some replication of the DNA. How many chromosomes then would I have?
56.
Yeah, I should have 56. Okay, so there's going to be replication going on here. And this is, of course, during the interphase portion, replication. And I've got 56. Then the division, that's going to be the prophase, anaphase, prophase, metaphase, anaphase, telophase. And then ultimately I'm going to end up with 2 daughter cells. How many chromosomes will each of those have?
28.
28 again, you got it. And then it stops, that's the end. Now a couple things to describe these. First of all, when I'm talking about the number of chromosomes here, remember I've got some words to describe numbers of chromosomes. What is the word that describes the normal amount of chromosomes? So do you remember?
Diploid.
Yes, that's right, diploid. Diploid, which diploid essentially means double half. Okay, so that means the normal amount. And sometimes it's referred to as just simply 2n. So here at 28, I've got 2n. And at the end, each one of these also has 2n, diploid amounts of chromosomes. And again, the n just means half, 2 means time, so it's 2 times half, diploid. Here at the 56, though, I have twice the normal amount, or 4 times the half amount. What is it?
Tetraploid.
That's right, we call that tetraploid. Tetraploid is going to be 2 times normal, or 4 times the half. And that would be maybe, we could call that 4n. So in this case it would be 4n. There's only a small instance where any cell in your body is actually 4n or has double the normal amount of chromosomes. And this is, of course, during the G2 phase and partially through mitosis. And that's it. Any other time it's going to have 2n or the normal amount of, chromosomes. So those are my somatic cells, my body cells. Let's divide that up. Let's look at the meiosis now in the gonad cells. So I've got 28 going on here. Once again, I'm going to see some replication here. Just like with interphase up above, replication. And then that's going to give me, again, 56 chromosomes. Now I'm going to divide once, and I'm going to ultimately end up with 2 daughter cells that still have the 28 from before. But there's something different. Something happens in between this 56 and the 28. What is that? That's right, the crossing over. So we've got crossing over going on here. Crossing over. Now let's talk about what crossing over is. Remember, crossing over, so I have a chromosome from your mom, and I've got a chromosome from your dad. During the replication process, first of all, what are these called? These chromosomes that are from each parent, but they code for the exact same thing, but just slightly different. What are those called again?
Sister chromosomes?
Close. It's the other one.
Homologous.
Homologous chromosomes, that's right. These are homologous chromosomes, as in they code for the same thing, just slightly different. One's from mom, one's from dad. When they replicate during the during the S phase, I'm going to end up with now, Alexia?
Sister chromatids.
That's it. These are the sister chromatids. So these two connected together from dad, they're exact copies of one another. Those are sister chromatids. These two here, one from mom, one from dad, those are homologous chromosomes. Right? So that's what those two words mean essentially. Homologous chromosomes are one from mom, one from dad, but code for the same things. Sister chromatids are exact replicas of each one of those chromosomes from each parent. Okay? So now what I've got is I've got 4 chromosomes, 2 exact replicas from mom, 2 exact replicas from dad. And what they do is they connect right here in the center at the kinetochore, and they kind of lay on top of one another like a bowl of snakes or spaghetti. They're just like ugh, all over the place. And then they transfer genes back and forth randomly, and it's a lateral transfer. Remember, it's not going to be from up here to down here or vice versa. It's going to be straight across. And one set of genes is going to replace the other set of genes. So if I were to zoom in and show this a little bit closer, here's a chromosome, here's a chromosome that's cut into these sections here, like this. So here is eyes, here is ear shape, here's nose shape. Again, same thing, eyes, ear, and nose shape. What's happening here is this section of eyes is breaking off of one chromosome, and it switches places with the other chromosome. So where this was from mom, this was from dad, this is dad's genes, this is mom's genes. Now what we're doing is we're shuffling them. So now it's still mom's genes, but now it has a little bit of dad, and this is still dad's genes, but it has a little bit of mom, because that one switched places. That's what the crossing over is. But remember, it's lateral, it goes straight across. I can't switch eyes with nose down here, because then this chromosome would have 2 noses, and this one would have 2 eyes. That'd be weird. Like, I don't need 2 noses. I smell cat poop enough at my house. I don't need to smell worse. So it's just going to be a lateral switch, back and forth. Eyes and eyes. Okay? And remember another thing, too. The closer those things are to one another, the more likely they'll move together as a unit. So eyes and ears are pretty likely to switch with one another, but eyes and nose not necessarily will switch with one another because they're so far apart. Right? So keep that in mind too. But it's going to be a lateral switch back and forth, and that's what crossing over is. Now when that crossing over occurs, shrink this down, get it out of the way. There we go. When that crossing over occurs, now I've got kind of a shuffled version of the chromosomes, right? And then I'm going to get another split here. Oh, I almost forgot. Hold on. Before I even get into that. One other big difference between the two. Let me go back to this. Almost forgot. So that crossing over happens during prophase 1, so at the very beginning of meiosis. Then after that, during the metaphase and anaphase, when they separate, when we're talking about mitosis, here's how they line up. So I've got my chromosomes like this. Here's from mom. Here's from dad. They line up along the metaphase plate right there in the center. And then what happens is those sister chromatids split apart. One goes to the left, one goes to the right. Okay, so notice how they're lined up there. In meiosis, instead, they line up next to one another. So here's mom, and here's dad. And they split up homologous chromosome style. Okay, so notice the difference between the two. Here, mom, dad split equally, and each cell gets mom and dad. Down here, the homologous chromosomes line up next to one another. And so one gets mom, one gets dad
So which
instead.
one?
Yes.
So the top one is mitosis?
This one's mitosis
Okay.
right along
Oh,
here.
okay.
And this one's meiosis right here. Draw that, finish drawing that one. There it is. Okay? So this happens during the first metaphase and anaphase of meiosis. So again, during mitosis, we're going to be splitting those sister chromatids in half, and then each cell is going to get one of each. Down here in meiosis, we're separating the homologous chromosomes. And so one cell's going to get mostly mom, the other one's going to get mostly dad. And this kind of results in a nice shuffling of the genetic material. Nice kind of like shuffle thing going on there. Move this out of the way. There we go. Once that has happened, once those chromosomes have split up, we're going to go through one more round of division. Does the cell replicate its DNA again for my meiosis and for that second round of division? No, it does not. It just immediately goes into another round of division. And when it does that, it's going to make now 4 daughter cells. But how many chromosomes is each one of those going to have?
14.
That's right, 14. And we call this,
Haploid.
that's it. So this was diploid, tetraploid, diploid. These are just simply N for haploid, or half. Haploid. Half. N. There it is. That's a 14, by the way, not a 44. What the heck I'm doing with my life? Just wasting it. There we go. Got it. All right, so I make 4 daughter cells. What do those 4 daughter cells ultimately become? What's the fancy term for them? Begins with a G, I heard it? Gametes. What is the layman's term for gametes? You got it, sex cells, like sperm and egg. There they are. Those are my gametes. Fun factoid about sperm and egg, I think we mentioned this before though. When sperm are made, all 4 of these gametes ultimately become sperm. But when an egg is made, only one of the gametes actually turns into an egg. The other 3 are discarded. And the reason for that is because the egg has to be really, really big. So as it divides, one of the 2 is always going to get more cytoplasm and organelles than the other one. So in this case, I would get this one really, really big, and this one would be kind of a little dwarf cell. And then they would still divide, just to finish the process. This one would divide, and then one of these would be a little dwarf, and the other one would get the majority of the cytoplasm. And ultimately, at the end, I have 1 large egg, and then 3 smaller cells that end up just being reabsorbed. And so that's why females tend to make a lot less eggs than males do. Grace Finetti. Denzel.
Why does it start with 28 chromosomes?
What's that?
Why does it start with 28 chromosomes? Like,
Oh, because I wanted you to show me that you understand how the division of the chromosomes works without having memorized the 46 to 92 and 46.
46.
What?
Will it always be like 46?
No, it won't always be. For humans, it will be. This is a different organism, though, that has 28 chromosomes. Remember, other organisms have different amounts of chromosomes, and so I wanted you to show me that you understand how they divide throughout this. So instead of just memorizing the numbers, I want you to memorize how the numbers get made. Got it? All right. All right, so let's talk about probability and Punnett squares, kind of like what we were doing last class a little bit. Do you remember with the Punnett squares? It's just simply 2 by 2, and I'm comparing 2 parents together. Remember, the capital letter represents what? The dominant allele, so the lowercase is perfect. Every single organism that gets made has to have has to have 2 alleles, but it can be a combination of how many, or like, I should say, I don't even know what that sentence was, but there we go. Let's look at probability, actually. So when I'm looking at this Punnett square, these are 2 parents that are both what we call heterozygous, because they have 1 of each allele, 1 dominant, 1 recessive. What is the probability that one of their children will end up with the recessive only gene? Right, 25%, that's this right here, 25%. Now that's their first child, 25%. And let's say they have a second child, what is the probability that that second child will also have little b? Still 25%, you got it. Okay, so that's the one thing that's interesting about the probability. It does not change regardless of how many children we have. You go all the way down to your 365th child, and it will still be 25% probability of getting little b. You're really playing the odds at that point. You're like really rolling those dice. But it is possible to still get one of the, like a little b. That's just how it goes. It's just usually the more offspring you make, the more those probabilities actually come out. And we'll see that in a problem a little bit later on. Okay? So we got big B, big B, big B, little b, big B, little b, little b, little b. So let's keep those in mind as we take a look at some of these pedigrees. Now remember, a pedigree is kind of like a family tree for a specific genetic trait. It can be a disorder, or it can be just a simple trait that you're trying to track. Okay? When an organism or when a person in the pedigree has that trait, they're shaded in. If you're a circle, you're female. Square, male. If you're open, you don't have that particular trait. So let's take a look at this entire pedigree here. And what it's asking is for us to determine the genotype. So which one of these 3 is this person at large B most likely going to be? Now remember, in order to figure this out, we got to kind of work backwards. So what we have to do is first determine, is this going to be a sex-linked trait? Now we talked before about sex-linked traits where we have the XX versus the XY. Females have two X's, so if one of those X's is, for whatever reason, has like color blindness on it, we'll use that as an example. You have a default X that you can go to instead. So this X will be ignored, this X will instead be used to code all the things that need to be coded for. So you end up having one that works. But here as a male, we only have one X chromosome. And so if that X chromosome has something on it, there's no other X chromosome to default to, and so we will always show that particular trait. What this means is that the majority of the time, if it's an X or sex-linked gene or disorder or whatever it is, that thing is only going to show through males. Females typically won't exhibit that. So looking at this pedigree, would you say that this is a sex-linked trait or not?
No.
And I would say no also. And the reason for that is because it's always showing in males and not showing in females. But I have two females who actually have this particular trait showing through, and a male. So because of that, not necessarily evenness, but I definitely have both sexes represented here, I definitely probably have a non-sex linked trait, which means, what would I call it instead? Does anyone remember the word? It begins with an A. A-U-T. That's it, autosomal. An autosomal trait. Autosomal traits can either be dominant or recessive. So now that I've determined that it's not sex-linked, that it's autosomal, now I have to determine if it's dominant or recessive. So you remember how to do that also. Let's take a look at this area right here, for instance. Is it possible for this to be a dominant autosomal trait and still have 4 children that do not exhibit that trait?
Yes.
No, it's not possible. Not if this was dominant, even just one dominant allele is present, that would mean that dominant allele would have to show up someplace. It's very unlikely that we would have 4 children that doesn't show that dominant allele at all. Okay, so I'm going to go ahead and guess that this is a recessive autosomal trait, which means that this person here would be little b, little b. And we'll fill out the rest. That'll be little b also, and this one too. Okay, now coming back to here, it wants to know what this person is. First of all, because they're not exhibiting the trait, which one of these 3 can we eliminate? A, B, or C?
C.
Definitely C. I can get rid of that, right? Which one are they most likely going to be, though?
B.
And why do you say B?
None
What?
of the kids have the little b recessive trait.
Right. So if I'm going to, and I'm going to explain this in just a second, but if I am going to do a Punnett square to show this, I've got big B, like we're guessing, and then we have little b. What I'm going to end up with is 100% of my offspring being heterozygous, but because the trait is recessive, I need 2 little b's for it to show through. None of my children will have that, and so there we go, all 4 of them. However, is it possible for us to get this with somebody who is big B, little b? And it is. Okay, if you look here, big B versus little b, I do still have a 50% chance of having offspring that do not exhibit this trait, but they are carriers for it. Okay, so again, remember, the genotype, or sorry, the genetic scaling, or the pedigree is not gospel. It's not 100% foolproof. It is totally possible for these 2 to be like this, big B, and little b, and have a 5th offspring. We'll make it male this time, and this one suddenly does have that trait. And what that does is it's going to completely reverse everything that we just discussed and make this person big B, little b, right? So it is all dependent on generation, and that's why you usually like to have multiple generations. It's kind of impossible for you to figure out who's big B and little b or whatever in your family if you're just going to look at you and your sisters and brothers. You've got to look at your parents and their parents as well, and look at anybody else related to you. Also, the more information you have, the better chances you have of figuring out what it actually is. So let's go to here now. Let's look at A here. What do you think the most likely genotype of person A would be? Again, take a look at their offspring. We already determined that this person here is little b. So what does that person have to be in order? Bless you. Yeah.
A. Big
That's
B.
right. It has to be. That's right. It has to be big B, little b, or letter A. And the reason we can determine that is because, again, from our previous our previous Punnett square, if I put a big B, little b, and another big B, little b together, that will result in at least 1 or 25% of chance of getting a little b, little b. Okay? And that kind of plays with those numbers pretty well here. Okay? Let's take a look at this one. All right, how about person C? What's the probability that person C is a genotype big B, little b? So now it's not trying to determine what the genotype is, we're saying that the genotype is big B, little b, what is the probability? So again, we need to figure out a Punnett square here. We need to know what these two parents are. We know that this one's little b, little b. We also know that this person here is little b, little b. And so if that's the case, that means that this parent has got to be, that's right, it has to be a big B, little b. It can't be a big B, because otherwise I would not get any offspring that have this particular trait showing through. So now what I want to do is figure out what is the probability that they are big B, little b. So in this case, we've got a big B, little b, and we've got a little b, little b. Oh, shoot. Oh, gosh. There we go. So what's the probability that that person is big B, little b? With what? Ooh, so now that is this is a good thing to look at now. This is kind of interesting here. Are they little b, little b?
No.
Then? They're not. So that kind of takes it out of the running. So now what's the probability that they're big B, little b?
100%.
It's 100%. It is a 100% probability that they are big B, little b, simply because they do not exhibit this particular trait. This is where the probability and statistics kind of comes in. It's kind of weird like that. Because these are not in the running anymore, these are the only options. Now, it would still be 50%, let's say, if we had, say, an option for this, and I don't know how this would be possible, but an option for that, and these were taken out. Now it would be the probability that they were big B, little b.
50%.
Now it would be 50%, because I know it's definitely not little b, but it could be either of those, and so 50% chance that it could be that. But obviously this Punnett square is not possible, so I'm just going to ignore that. So we've got a 100% chance that this person is big B, little b. That's cool. Let's go ahead and fill that in here. How about letter D? What about that person? What could they possibly be here? Well, sorry, what is the probability that they are big B, little b? My bad. What is the probability that they're big B, little b? Okay, so remember, we've got this, and we've got this, right? This is what we determined these two to be, because that's what would result in this. This person is definitely a little b, little b. We don't know 100% what they are, but let's do a Punnett square and take a look.
So 75.
All right, so I have a big B, big B, big B, little b, big B, little b, and then little b, little b. Which one of these four are they definitely not? They're definitely not that, okay? So what are my options? I've got a big B that they could be, and then I have two big B, little b. So it's basically what?
66.
Right, 66 or 2 out of 3, right? So there's a 100% chance of that, and then I've got a 66% chance of that. Okay? Yes?
So these questions, you're answering what it would be without risk. You're not accounting for the smaller one, or what it can't be, for sure.
Right, exactly. You remove that because you can physically see that that's not one of the things. So you're
Oh.
only looking at what could possibly be. So
Alright.
it can't be this. It has to be one of these three. So what's the probability that it's those two out of this one? So
Okay.
that would be 2 out of 3, or 66%. Now, just FYI, these types of questions aren't going to be on this test. I just thought this would be kind of interesting for you guys, since you guys seem to be like actually receptive to learning. So that's cool. So one more, and this one's really kind of cool. All right, so we determined that this person was big B, little b, and that was 100%. We determined that this person was possibly big B, little b, and that's a 66% chance. So now the question is, what is the probability that these two people will have a child that has this particular trait? So, what does their genotype have to be in order for them to have the trait? Little b, little b. All right? So, let's go ahead and put together Punnett square. Check this out. So, we've got big B, little b, big B, little b. So, we've got a big B, big B. We've got a big B, little b, big B, little b, and a little b, little b. What's the probability that these two will have a child with the disorder, or with this particular trait? 25%, okay, or 1 out of 4. However, what was the probability that this person was going to be big B, little b? That's 100%, so that's a 1. What's the probability in a fraction that this person would be big B, little b? So I'm going to multiply that by 2/3. And then, what's the probability that these two will have one of those? It's 1/4.
Oh.
So now multiply all that together. What is it? Yeah, it would be 2 over 12 or 1/6. So there is a 1/6 chance, or a 1 in 6 chance, that these two people coming together will have an offspring that will have that particular trait, whatever it is. That's cool? That's how probability and Punnett scores essentially work together. I thought so. Okay. I did have one other thing for us, but we just don't have time to do it, so we're not going to do it right now. But I will save it for next time, maybe, so we can go over it together. Or I might post it on Google Classroom with like a video showing you how to answer it. Either way, there is going to be an FRQ on the test, FYI. It won't be nearly this long, but it will be an FRQ to study for anyways. I'll give you a hint. It has to do with DNA and that thing about the negative stuff on us, how the DNA is negatively charged. But that's all I can tell you. That's it, okay?
DNA negative.
And that's it. The end. Now don't forget, on Classroom, I have a couple things for you to use to help review. First of all, the study guide is a doc. I will be posting the answers Saturday for you to compare your answers to. So my suggestion, get this done before Saturday. You know, you don't want to do that thing where like you answer it all afterwards and you're just looking at my answers. You're like, I would have put that anyways, that's right. Also, I've got the review homework 004 if you haven't done that yet. And my suggestion, do the other review homeworks too. Just good study and practice. Whatever. Okay?